All right, so talking about cellular respiration, the first step to cellular respiration, what we're going to look at today, is called glycolysis. So this is our step number 1 to the 4 steps of cellular respiration. So there's a few things that we should note. First is that it occurs within the cytosol of the cell. So that's that liquid component of the cytoplasm, which consists of ions, water, and small molecules. So it occurs in the cytosol of the cell. Another important note that we should make is that there's no oxygen that's required. So no O2 required. So it's anaerobic because we're not using oxygen, and almost all organisms will undergo glycolysis. So the location is in the cytosol, and there's actually no oxygen required, meaning that almost all organisms will undergo glycolysis. So we're going to do 5 steps. So this is actually the first 5 steps of glycolysis, and then we'll move on to the last 5 steps. So the first step, we're going to take a glucose molecule. So before I do that, I'm just going to make a little legend of each color that I'm using so it's clear to everyone what each color actually means. So I'm going to use green for the steps and enzymes that are required throughout the reaction that we're going to see. So green will be for enzymes and steps. I'm going to use pink, and this is just going to be other reactants and products that we see. / products. And then lastly, I'm going to do red for our phosphate groups. And this is what we're going to see through the substrates that we draw out. Okay, so those are the colors that I will be using. So for step 1, we're actually going to start off by drawing that glucose molecule. So we're starting glycolysis off with a glucose molecule. So this is a 6-carbon molecule. There's an oxygen group there. And then we have a CH2OH here. So there's our glucose molecule. We're going to name it glucose. And then we're going to do step 1. So again, I'm going to outline the steps in green. And the enzymes are as well. I'm going to represent those steps that we're going to go through. So what happens first is that glucose needs a boost of energy to get this reaction started. So it's actually going to take ATP. So ATP comes in. It's going to donate its phosphate group, thus turning into ADP. So it just lost a phosphate group because it's going to add a phosphate group onto this next molecule that we're going to draw. So step 1, glucose needs a boost of energy. ATP comes in and donates its phosphate group. This then creates another molecule, which we're going to draw out again. This is also a 6-carbon molecule, but the only difference is that we're attaching a phosphate group onto this CH2OH. So again, we'll draw our molecule. So there's our oxygen. And then here you would do your C2OH. But here's the difference. We see that phosphate group has been added here. So this is now a new molecule because we have added a phosphate group. The phosphate group was added onto the 6th carbon. Thus, this molecule is called glucose 6-phosphate. And it's important to outline which enzyme actually facilitated this reaction. It's called hexokinase. So that's step 1. All right, so moving on to step 2, again, outlining in green. So what happens in step 2 is that we are going to take an enzyme. It's called phosphoglucose isomerase. So through the name of phosphoglucose isomerase, you can predict that we're actually going to be turning this glucose 6-phosphate into an isomer. So an isomer is exactly the same molecule. We're just rearranging the shape. So our oxygen's actually going to be at the top. Then we'll have our CH2OH. And then we're gonna see this CH2OH with its phosphate group attached here. So we have just created an isomer. This isomer is called fructose 6-phosphate. So again, we added this enzyme phosphoglucose isomerase, which turned our glucose 6-phosphate into this isomer of fructose 6-phosphate. So there is our second step. We're then going to jump to our third step. So our third step, we need another boost of energy. Where do we get that boost of energy? We get it from ATP. So ATP comes in once again. It donates its phosphate group, thus turning into ADP. So we're going to redraw our new molecule that we have. Our oxygen's still at the top, but this is where we see a difference. CH2OH, we're actually going to add another phosphate group here because we just donated phosphate from ATP. We're then going to redraw our CH2OH with our phosphate group. So we now have 2 phosphate groups on this new molecule. Our phosphate groups are on carbon 1 as well as carbon 6. So this means that this must be called fructose 1,6-phosphate. So there's our new molecule because we added a phosphate group from ATP. And it's important that we label which enzyme facilitated this. It is called phosphofructokinase. So there's our step 3. So step 4 and 5 are interesting because what we're actually going to do is we're going to take that molecule that we just drew, this fructose 1,6-biphosphate, and we're going to split it. So this is a 6-carbon molecule. So we're going to split a 6-carbon molecule into 2 3-carbon molecules. So I'm just going to draw 2 arrows here because this 6-carbon molecule is going to be split into 2 3-carbon molecules. So the first step, so we'll do 4 here and then 5. So our fourth step, we have this enzyme coming in. This enzyme is called aldolase. So aldolase, what aldolase is going to do is it's going to split our fructose 1,6-phosphate into 2 3-carbon molecules. So let's draw our first one. So we have a phosphate group at the end here. We then have an oxygen attached with a CH2 attached. We then have a carbon with a double bond oxygen, and then we have our CH2OH. We're not drawing the molecules like this anymore. We're just going to do them like this just to make it a bit more simple. So this new product that we just drew is called DHAP. It's also called dihydroxyacetone phosphate, but we're going to call it DHAP. And then the other molecule that we create through the use of aldolase is called G3P. So we have our hydrogen at the top. We then have a carbon with a double bond oxygen, CHOH, and then we are having our phosphate group on the bottom here. So there's that phosphate. So in step 4, we took fructose 1,6-phosphate, aldolase, split this 6-carbon molecule into 2 3-carbon molecules, 1, 2, 3, 1, 2, 3, and it created these new molecules of DHAP and G3P, also known as glyceraldehyde 3-phosphate. But then we have one final step, step 5. So what happens in step 5 is that another enzyme comes in. This enzyme is called isomerase. Isomerase takes our DHAP molecule, which we just created, and creates another G3P molecule. So isomerase takes our DHAP and creates another G3P molecule. Again, isomerase, this is an isomer of DHAP. So we just created another whole molecule in step number 5. So we actually end up with 2 G3P molecules. This DHAP molecule is just an intermediate step within the process of glycolysis. What happens with the DHAP molecule is isomerase comes in, creates an isomer, which happens to be another G3P molecule. So that means that we have just ended the 5 steps with 2 G3P molecules. This is very important. 2 G3P molecules have been created at the end of step 1 to 5. So it's important to also note that this entire process is called the energy investment phase because we used ATP. We needed to use some energy for this whole thing to happen. So this is called the energy investment phase. So I'm just going to do this. This is the whole reaction. It is called the energy investment phase. We're using ATP. Okay, so in steps number 6 to 10, those 2 G3P molecules are going to be the beginning molecule that we see in steps from 6 to 10.